The Institute for the Study of Peak States presents the second International Psychoimmunology and Psychobiology Research Symposium. The Epigenetics of Trauma, a New Paradigm for Trauma Healing, presented by Dr. Celine Guerin.
Okay, so our next presenter is Dr. Celine Guerin. She's French. She has a PhD in neurosciences and was trained as a peak states therapist about 6 years ago. After a PhD, she worked in psychiatry and explored different alternative healing practices before discovering peak states therapy. She is now receiving individual clients for peak states therapy in Sophia Health Centre in Lausanne in Switzerland, but also worldwide with online peak states therapy sessions. She specialises in stress management, burnout, sexual abuse, and self-esteem issues. She's part of the Institute's applied research team and delivers training in spiritual emergency for our students. So over to you, Celine.
Thank you. Hello. Hello, everybody. I set up the... Okay. So today I will speak about epigenetics of trauma. Probably you heard about trauma and probably you heard about epigenetics, but you might not know how this affects your daily life. So my talk is about, is like an introduction to the model of psychobiology developed by Grant McFetridge for the last 30 years. And this model of psychobiology, it's not only explaining that emotion can affect our body, actually it's the opposite. This model is showing that there is something already in our body that will create our emotions and that will affect the way we feel. So more than an understanding, this biological model is bringing us some new tools, very efficient and fast tools for trauma healing and to change the way we feel. I guess living a fulfilling life is something that everybody is looking for. Personally, when I was young, I was thinking, yes, I want to be happy. This is my aim in life. And soon I realized it's not that easy. I don't know, I could give some example, like I had some friends and at some point I could feel not understood or lonely or angry. And I was always thinking, what make me suffer and what make us suffer in general? And that was a quest that stayed with me all along my my studies and my personal way. So at that moment, at that time, it was 90s and genetics was considered like the answer for many things. We were thinking that if we know, if we can sequence our genes, that we will have answer for many disease and many behavior and many psychiatric disease. And finally, it was not the case. And on the way, we discovered something else that's called epigenetics. And this is giving an opening, thinking that maybe we have free will and what is the part that we can choose, what make me be what I am today and behave the way I behave today and the way I feel. And this epigenetic for me was the key. The first time I heard about this at university, they used to call this junk DNA, and I was thinking how nature could create and keep so many useless DNA in our body. And I was thinking probably here there is a key to understand how we can have this free will to change and to decide for our life. And this is exactly what psychobiology is explaining and is understanding, and propose some therapy to increase our inner potential physically, but also emotionally, and in terms of brain functioning and acting on metabolic pathway. And this is what I will explain you today. Let's talk about homozygote twins. Maybe you've seen some of them and they are very similar in the beginning of their life. And sometimes they are starting to differentiate and to have different behaviors, different taste, even different look, more and more old they get. So how can we understand this? Because they have the same genetic background, but then they can develop different phenotype. And some of the later studies have explained and have shown that two twins, for example, one stayed on Earth and the other one went into the space, and they've seen all the changes that happen on epigenetic level. And some studies have shown also the link with the apparition of some diseases. So again, they have the same genetic, but they can develop different disease, they can develop different behavior, depending of what they eat, depending of their lifestyle, if they do sport, if they smoke, or if they are exposed to pollution or to different kind of stress. So what is epigenetics? There is 3 types of epigenetics that are quite well described today. And epigenetics is all the chemical modification that will change gene expression without changing the DNA sequence. This modification could be like DNA modifications, methylation, or we could have modification on the histones. The histone is the protein that will make the compaction of the DNA. And there is a third one that we call the non-codant RNA. I will not go into details, but it's to explain that those epigenetic modifications are stable, heritable, and we can reverse them. And this is what is really interesting for us. Now, in science, we use animal model to study, for example, stress and psychiatric disease or other physical disease. And there is some model for anxiety, because anxiety is the most common psychological disorder. Actually, 1 in 5 persons will be affected once in their life. And it's a huge cost for the governments. So of course, there is a lot of studies around that. And this is the synthesis of many studies, but just to show you that what we do with mice, we create what we call lineages of mice with the same genetic background by making them reproduce between brother and sister. And after that, we have those mice with the same genetic background, like the twins. And then we can do some experiments on them to check the influence of epigenetic. So for example, they separate the babies from the mom. And what they figure out is that the mice after will present more anxiety. So in the first case, when there is no exposure to stress, the mice go back and forth in the black chamber. And when there is the stress that is induced in the baby, this one will show more anxiety and it will go much less to explore the dark chamber. And a few years later, I show you this article, because for me it was like, wow, quite a revolution. And not just for me, it's an article that was published in Science, and that is showing that maternal care will influence epigenetics and also acquired behaviors. So it's the same kind of model. And in this case, they are taking 2 different lineages, so same background, but different epigenetics and different behavior. So one behavior is the mom who is a bit neglecting the babies, and the other one is like the normal one, let's say, and where the mom is really taking care of the kids, licking them, grooming them. So what they know is that there is different epigenetic patterns for those 2 lineages. And what they did cross-fostering between the 2 lineages. So 2 babies from the neglecting mom were put with the caring mom, and the opposite. And what they have seen is that those babies, when they are raised, or when this mom, the caring mom, is taking care of them, they will be able to change the epigenetic pattern and to learn this behavior, and the opposite for this strain. So they are getting more stressed, even if they are coming from this biological mom who is able to do the leaking and grooming. So they lose this ability and they change in the epigenetic marks. So it means epigenetic marks is related to the resistance to stress. They can be acquired at the same time as the behavior is learned. And it could be, this example, mediated by maternal care or any other environment. And another thing that they have shown in this study is that this is transmitted to the next generation. So if they lost the behavior or if they gained the behavior and the epigenetic pattern, then this could be transmitted to the next generation. Now let's go in human. We know for a long time with psychology that trauma are involved in stress response. Actually, trauma is an emotional response to a terrible event when we are confronted to death or risk of death for ourselves or for another person, like an accident, rape, natural disaster, war, terrorist attack. And some people will develop what we call post-traumatic stress disorder, when they have long-term reactions, emotions, flashback, nightmares. This will affect their relationship, and they can even show some physical symptoms like headaches, nausea. And what psychoanalysis is adding is this notion of non-acceptance. It means the person who is going through this kind of shocking event will not be able to accept it. And the memories keep coming. What we know also is that any kind of adverse childhood experiences like mistreatment, violence, abuse, neglect will increase the risk to develop this post-traumatic stress disorder, but also other mental disorders like depression, anxiety, and addictions. The question is why some people will develop PTSD, so this post-traumatic stress disorder, and some people are more resilient. What could explain that? Let's continue with a few scientific studies. So those studies have shown that trauma also affects epigenetics. Actually, an increase in severity of the abuse in childhood results in an increase of number of methylation. They have shown this like taking several psychiatric patients, and they have shown in them that depending on the number of abuse they went through during their childhood, they would show this increase in the methylation modifications. And a study from the same group a few years later have shown that those methylations related to childhood trauma are correlated to PTSD, to depression, and also borderline disorders in adults. So now we have the link between what we are going through during childhood, how we feel as an adult, and what kind of psychological disorder we can develop, and epigenetic marks. What we have shown also, what we have seen in some studies, for example, during the 9/11 events, they have studied the mom who were pregnant at that time and close to the World Trade Center. And what they have seen is the mom who were going through a PTSD, so this post-traumatic stress disorder, would have kids who would have an increased response to stress in the first year of life. That they didn't study after, in this study at least. And so we know that even when the baby is in the womb, we can see that there is effect on the stress reaction as a baby and probably later on in life. Actually, the first study started with the children of Holocaust survivors. And this is when we first were thinking that there is a correlation between some trauma in the parents and what the kids will develop later in life. So for example, those children from the survivors are presenting more risk to develop PTSD, anxiety, and other mood disorder again. And it's not just about emotional trauma. Actually, what science is showing is that famine, like starvation, in China caused increasing in diabetes type 2 in population. And those children whose mother were pregnant during the famine in the Netherlands shows specific epigenetic patterns. So again, we were able to link an event that happened when the kids were inside the womb of their mom and what's happened in terms of physical disease later on in life. So trauma during pregnancy and early childhood, like stress, violence, and starvation, will affect epigenetic pattern and are related to physical problems or psychiatric disorders. And we can see that they affect the next generation. So for many years, in conventional psychology, we were thinking that trauma, this is the mark I put here, let's imagine that this is all the trauma events the person went through. And we were thinking the trauma starts just at birth, you know. But finally, what science, the latest, the last year, the last 10, 20 years, 15 years, they started to show that even what's going on when the baby is in the womb of the mom can affect his life. And what our model is saying is that things doesn't start just here in the eggs and sperm, but it starts even before. And the eggs is actually, it's not formed, it's coming from the primordial germ cell in the maternal grandmother, and the sperm is coming from the same germ cell in the paternal grandmother. So actually, even some trauma that could happen here would affect the kid and then the adult. So it means it's not, in science, usually what's happened during the life of our parents is called transgenerational transmission. For us, this is still the biographical trauma. This is a big difference that our model is bringing. And let's go back now, let's go to this lifetime. So with the same trauma, and what our model is explaining is that it's like if there is a repetition. And one trauma string, this represents a trauma string. I will show you after what is it about. It's mRNA actually. And on this mRNA, it's like if some of the same emotion and the same physical symptoms are like stored in those dots that represent actually ribosome. And let's imagine this is two different trauma strings with two different types of emotions and different physical symptoms. And you can see, let's imagine this is, for example, a car accident, and the person will develop a PTSD after that. And we can see, so this will be the event that activated or triggered PTSD. This is what we think, but actually in psychology. But actually, in our model, what we would do is to go back in the past, because probably there is some other trauma before with the same emotion and the same physical symptoms. So the origin of the trauma would not be here. And here is another trauma string, for example, that is linked with other trauma events. So the thing is, the trauma could start here, or the trauma could start after the conception, or even we could have trauma after birth. But very often we have to go back in the past till the grandmother. So this is our trauma string. In our model and in our therapy, what we would do, we would bring back the person in the trauma moment, for example, the car accident. And we would evaluate the SUDS, the subjective units of distress that the person feels here, find the emotion, find the physical symptoms, and then we would do the regression all along this trauma string to find the first trauma moment. I'm quite amazed how people are able to do that. And some people even have some visual information or where they are. And for some of them, they don't have this visual information, but doesn't matter because they are almost all able to do this and to heal this first moment so linked with the damage of an install, and just doing the 1-point tapping until the person is feeling calm, peace, and light. And at that point, it's like if you release all the trauma string. And in our model, this trauma string is in a cell that we call the primary cell. And it's like all the ribosome are here stuck because of the damage of the histone. So trauma moment would be a moment when the body needs a specific protein to act or react or just continue to feel calm, peace, and light. But as the body doesn't have this protein, then we will have an emotions, and this will create a memory as a frozen image. And in this model, the PTSD would be the extreme cases of biographical trauma. But actually, this model is the same for other kind of emotions, sadness, loneliness, or anxiousness, stress. Any other kind of emotion could come from a biographical trauma. And very interestingly, one of the group of research have shown that RNA could mediate the memory of trauma. Trauma could be stored in RNA itself. So what they did, they give electrical shocks to snails. They took the RNA, they injected to the snails, and they observed that those snails that were not trained, not sensitized to those electrical shocks, they got the same stressed answer that the snails who received the electrical shocks. And also what they have shown is that if you put the RNA on sensory neurons in culture, this will increase the excitability of neurons. So this is quite amazing. It means really probably the trauma are not stored only in the synapses and in neurons the way we were thinking before, but probably they are more stored in RNA itself. So this research provides new clues in the search for the physical basis of memory with possible application in Alzheimer, and tends to confirm our psychobiological model of mRNA trauma string. So, of course, now it would be very interesting to be able to prove that therapy repairs epigenetic damage. And we have some ideas about how to do that and how to measure epigenetic marks before and after a biographical trauma treatment, for example, using this kind of microchip where you can check all the methylation pattern. And so we could have look if it's about one or several methylation changes that are happening, and if we actually make those changes after the treatments. So we have several projects that are going on. One would be with women in Kosovo who were pregnant during the war. So I'm working with the NGO, and now we are looking for financial funds to be able to go on with those studies. But we could also use any kind of PTSD patients, like for example, people after terrorist attack who develop PTSD after the attack in Bataclan. There is many other examples that we could with who we could work with. So this psychobiology is really a new paradigm to psychiatry and medicine for me. It was really a revelation when I discovered the work of Grant and all the institute. And here I just introduce you to and talk about biographical trauma, but actually the model is talking about much more than that. We are describing generational trauma, body association trauma, and many other subcellular cases. You will see in the next 2 days many applications of subcellular psychobiology, and you will hear just after my presentation about psychoimmunology that is really promising for curing a lot of disease. So in conclusion, emotions are based on conscious and unconscious trauma. It's not just about war, terrorism, and abuse. This really concerns everybody. And psychobiology is giving us fast and efficient techniques to heal trauma. So we all have the possibility to have a better life with more peace, more joy, love, and success. Thank you very much for your attention. Now, if you have any question, I'm ready to listen and answer to them. And if you have any question about research or any possibility to find funds for the research, please feel free to contact me at my email address. And I will finish with that quote of Carl Gustav Jung that I really appreciate. And we're saying, what is not brought to consciousness comes to us as fate. Thank you.
Thanks, Celine. There is one question here, but it's a very long one. So I'm just trying to work out how to quickly summarize it. So it's from Antonella Ercolani. She's saying, from the little that she knows, that some illnesses are turned on by damaged epigenetic markers, methyl or histones, and the proteins are not released. And this is where trauma healing comes in. And the proteins are able to be produced and released. I think they may be not released. Other illnesses instead seem to be the product of a lack of regulation of genes that don't have the coding needed and are so turned on instead of being turned off. Okay, so you're following? So does the trauma healing work on this as well, directly creating the coding or clearing the way for other proteins to create histone codings for these genes, or indirectly with some other processes needed?
So if I would know about which illness she's talking about, maybe I could also have a bit more information. I don't think we are really able to say how it acts, what's happened. I think there is some okay, about methylation. There is also some reaction that's going on in the cell that will create an inhibition of the gene expression. So it really depends of which disease and what is the mechanism. But I think for all the disease, we are looking for the specific mechanism involved in it to try to find a way to solve it.
Thank you for watching. For more information, visit pigstates.com.